Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss gluconeogenesis. In previous videos, we talked about the postprandial state as well as the early starvation state. Humans do have another physiological state, and we call this late starvation. And it's important to understand late starvation so that we can understand prolonged starvation, the disease diabetes mellitus, as well as understand what happens when a patient is on special diet. As we emphasized in previous videos, one of the most important questions that we need to answer irrespective of whatever state we are in is what will maintain our blood glucose at least greater than 40 milligrams per deciliter. That's the critical value. So what maintains our blood glucose? Because this blood glucose is very, very important for your brain cells, which rely very heavily on blood glucose for their energy needs, as well as for your red blood cells, which can only use glucose for their energy needs. If you had your dinner, let's say around 6 p.m., so as you are eating, the glucose from your diet is going to enter your bloodstream through the enterocytes. Your blood glucose level is going to rise. It's going to increase, and that increase in blood glucose is going to stimulate insulin release. And as insulin is released by the beta pancreatic cells, it will stimulate the uptake of glucose, especially by the adipocytes and your muscle cells, and that will gradually lead to a fall in the glucose back to the reference range about 3-4 hours after your meal. So let's say around 10 o'clock or so, you go to sleep, your blood glucose is in the reference range and is slowly going down. And now what maintains your blood glucose? Because now you are in the early starvation state. And as we saw in the previous videos, it is glycogenolysis from the liver. The liver glycogen is broken down to glucose, and that glucose is exported into your blood, and that glucose will maintain the blood glucose levels around the reference range in early starvation. So as the glycogen in your liver is being used up to maintain your blood glucose, another process also begins to help maintain your blood glucose, and this is gluconeogenesis. So gluconeogenesis also starts occurring in early starvation, and if we still do not eat, if we get into late starvation, and you see that gluconeogenesis is very, very important to maintain your blood glucose when you enter late starvation. Besides the increase in gluconeogenesis during the starvation state, as starvation continues, another process becomes important, and that is lipolysis, the breakdown of triglycerides into fatty acids. And you will see that the other tissues, such as your muscles and your adipocytes and your liver, so except for the brain that uses a little bit of fatty acids, but mostly still glucose, and the red blood cells, they use only glucose, you will see that fatty acids will be very important in providing energy, ATP, for the other tissues during starvation. We discuss lipolysis as well as fatty acid beta oxidation under early starvation in the previous videos. So today let's look at this process here called gluconeogenesis. This process begins in early starvation, and by late starvation it becomes a very important process to maintain our blood glucose level so that we can continuously feed the brain as well as the red blood cells. Gluconeogenesis can be carried out by the liver, which is a huge organ, weighs about 1 kilogram. It can also be carried out by the kidneys, but only by the proximal tubular cells in the kidneys. And remember, the kidneys is much smaller than the liver. Each kidney weighs only about 100 grams. And more recently, it has been reported that the enterocytes in the small intestine can also carry out gluconeogenesis to a small extent. As the name suggests, gluconeogenesis means the creation or the synthesis of new glucose. Comparing gluconeogenesis and glycolysis is a great way to understand the difference between catabolism, which is what glycolysis is, a catabolic pathway, as well as anabolism, which is what gluconeogenesis is. So remember, metabolism is a combination of catabolism and anabolism. Glycolysis occurs in every cell, while gluconeogenesis, as we mentioned, occurs in hepatocytes in the liver, the proximal tubule cells in our kidneys, as well as the enterocytes in the small intestine. Glycolysis is a catabolic pathway where a large chemical is broken into smaller chemicals. In case of glycolysis, the large chemical is glucose, which is broken into two smaller chemicals, pyruvate. Gluconeogenesis, on the other hand, is an anabolic pathway. An anabolic or synthesis pathway is one where smaller chemicals combine to form a larger chemical. So in case of gluconeogenesis, two small chemicals, two pyruvates, combine to form one glucose. Catabolism involves breaking carbon-carbon bonds, and we see that in step number 4 here, where one intermediate is broken down into two intermediates by the breaking of a carbon-carbon bond. On the other hand, in synthesis, in anabolic pathways such as gluconeogenesis, we are forming carbon-carbon bonds. Okay, once again, we see that in step number 4, where two small molecules, dihydroxyacetone phosphate and glycerol 3-phosphate, combine form a carbon-carbon bond to give us this fructose 1,6-bisphosphate. In catabolism, we require oxidizing agents. So in case of glycolysis, the oxidizing agent is NAD. In synthesis or in anabolism, we require reducing agents. So in case of gluconeogenesis, the reducing agent is NADH. So basically here we are taking hydrogens away from the carbon in catabolism. In synthesis, we are adding hydrogens to the carbon. Catabolic pathways such as glycolysis release energy in the form of either ATP or GTP, while anabolic pathways such as gluconeogenesis require energy in the form of ATP, typically ATP, sometimes also GTP. So in glycolysis, the glucose is broken down into two pyruvates. Now, to synthesize this glucose, we cannot simply take the pyruvate and make it into glucose, because recall that in glycolysis there are 3 reactions. Reaction number 1, that is irreversible. Reaction number 3, that is irreversible, as well as reaction number 10 that is irreversible. So we cannot simply take pyruvate and reverse glycolysis to give us glucose. So due to these irreversible steps 1, 3, and 10, for gluconeogenesis we need to overcome these irreversible steps. So step number 10 is overcome by two enzymes, pyruvate carboxylase and PEP carboxykinase, while step number 3 is overcome by the enzyme fructose 1,6-bisphosphatase of gluconeogenesis, and step number 1 of glycolysis, that is irreversible, that is reversed by using the enzyme glucose 6-phosphatase in gluconeogenesis. So basically, gluconeogenesis can begin with pyruvate. The pyruvate is converted into oxaloacetate by this enzyme pyruvate carboxylase, and you can see it requires energy here, ATP. The oxaloacetate intermediate, this reaction happens in the mitochondria. So the oxaloacetate in the mitochondria has to be transported out into the cytoplasm. And once the oxaloacetate is in the cytoplasm, then the enzyme PEP carboxykinase, which requires GTP, will convert oxaloacetate into PEP, phosphoenol pyruvate. PEP is then converted to 2-phosphoglycerate. This is a reversible reaction, so it can go in either direction. 2-phosphoglycerate is then converted to 3-phosphoglycerate. 3-phosphoglycerate is converted to 1,3-bisphosphoglycerate, and once again here we need ATP. And then the 1,3-bisphosphoglycerate is converted into glycerol di-3-phosphate, and for this process we need the reducing agent NADH. So the two glycerol di-3-phosphate that are formed from two pyruvates here, one of this glycerol di-3-phosphate will be converted to dihydroxyacetone phosphate by reaction number 5, and then one dihydroxyacetone phosphate and one glycerol di-3-phosphate will combine by reaction 4, which is reversible, to form fructose 1,6-bisphosphate. So the next reaction, recall reaction number 3, is irreversible. So for this we need another unique gluconeogenesis enzyme, and this enzyme is fructose 1,6-bisphosphatase, which breaks down fructose 1,6-bisphosphate into fructose 6-phosphate, releasing inorganic phosphate. This fructose 6-phosphate is then converted to glucose 6-phosphate using the same glycolysis reversible reaction number 2. And then we hit once again an irreversible step here for glycolysis, reaction number 1. So in this case we need another enzyme, and this is glucose 6-phosphatase, to convert the glucose 6-phosphate into glucose. So recall that glucose 6-phosphatase is present in the liver, not in the muscles, and this enzyme we did discuss under glycogen catabolism, glycogen breakdown. So to summarize, gluconeogenesis is a process by which we can make glucose from simple raw materials such as pyruvate. And for gluconeogenesis, we do use quite a lot of enzymes that are similar to glycolysis, but we must have 4 unique enzymes that are for gluconeogenesis that are not used for glycolysis. And these 4 enzymes are pyruvate carboxylase and PEP carboxykinase to reverse step number 10, to overcome step number 10 of glycolysis. And then we have fructose 1,6-bisphosphate to overcome the irreversible step 3. and then glucose 6-phosphatase to overcome step 1 of glycolysis. So besides these 4 enzymes, what else do we need to make new glucose? Since we are making new glucose, we require some sort of building blocks, carbon skeletons. So we will see that these building blocks could be pyruvate. It could be anything that can be converted into pyruvate, or it could be some sort of gluconeogenesis intermediate. For example, oxaloacetate is an intermediate. Or it could be anything that can be converted into a gluconeogenesis intermediate. So let's look at the major building blocks that these cells use for gluconeogenesis. So one is glycerol. Remember, glycerol comes from triglycerides. When we break down triglycerides, which are rich in adipocytes, you get fatty acids and glycerol. Now this glycerol can be transported to the cells that make glucose, such as liver cells, and there glycerol kinase converts the glycerol to glycerol 3-phosphate, and then glycerol 3-phosphate dehydrogenase will convert the glycerol 3-phosphate into dihydroxyacetone phosphate. Now, dihydroxyacetone phosphate is an intermediate of gluconeogenesis, so we could use this to make new glucose by gluconeogenesis. Another building block for glucose synthesis is lactate, which comes from red blood cells as well as several other cell types in the body. Lactate can be converted by lactate dehydrogenase into pyruvate, and this pyruvate can then be sent into the mitochondria, and the pyruvate can be converted into glucose by gluconeogenesis. So besides glycerol, lactate and pyruvate, glucogenic amino acids, so amino acids that can be made into glucose, and these are basically 18 out of the 20 amino acids, as we shall see later. So many of these amino acids that you find in proteins can be converted into pyruvate, or they can be converted into oxaloacetate, or they can be converted into something in the Krebs cycle that can be made into oxaloacetate. So all of this can give us new glucose by the process of gluconeogenesis. So during starvation periods, wherever the liver as well as the kidneys or the small intestine, wherever they can get their glycerol, their lactate, their pyruvate, as well as their glucogenic amino acids, all of this will be very helpful. They can use these raw materials to synthesize glucose and then send this glucose out into the bloodstream to maintain your blood glucose at more than 40 milligrams per deciliter, so that your brain and your red blood cells can be fed during this starvation period. Now, gluconeogenesis is an anabolic process. It's a synthesis process, and synthesis requires energy. So you can see, to make one glucose here from 2 pyruvates, we need 4 ATPs plus 2 GTPs. So that's equivalent to 6 ATPs worth of energy. And so where does this energy come from? And we'll see that during starvation, lipolysis is a predominant pathway, and when triglycerides are broken down, they give you fatty acids, and those fatty acids will undergo beta oxidation that we talked about in a previous video, and that beta oxidation gives you acetyl-CoA that can give you ATP, gives you FADH2 and NADH that can also give you ATP. So all of these things can enter the main street, your Krebs cycle, electron transport chain, oxidative phosphorylation, and generate the ATP necessary for gluconeogenesis in the liver or the kidneys or the small intestine. Besides energy, we do need some sort of reducing agent, in this case NADH. Now once again, beta oxidation of fatty acid releases a lot of NADH. So these NADH can be used as reducing agents for gluconeogenesis. So to summarize, gluconeogenesis, in order to make new glucose, the cells do require special enzymes, 4 unique enzymes that are necessary for gluconeogenesis, and we find those enzymes in the liver, kidneys, as well as the small intestine. We do need some building blocks to make new glucose. We do need reducing agents, NADH, and we do need energy. And these energy as well as NADH come from fatty acids, while the building blocks comes from several sources such as glycerol, lactic acid, pyruvate, as well as the glucogenic amino acids. So in the next video, we shall now look at this starvation story. So now that you know what we need for gluconeogenesis and what is gluconeogenesis, we shall now see what happens as we go from early starvation to late starvation and even all the way to, maybe this is 20 days here. What happens, let's say, after 50 days in prolonged starvation? Okay, so until next time, have a wonderful day. Thank you once again for watching, and hopefully we'll see you soon in the next video.